So this is part 4 of chapter 8, and hopefully the last part. So enzymes and temperature. Different enzymes function in different organisms in different environments. So we were talking before about how enzymes in humans um, usually have an optimal temperature of 37 degrees centigrade, and so there's a range, a small range, that they'll function at optimally. And you can tell what the optimum temperature is when you're given a graph like this by looking at where is the high point. So for hot spring bacteria or enzymes that are found in these types of bacteria, you can see the optimal temperature is actually 70 degrees centigrade by looking at this chart. Each enzyme also is going to have an optimal pH. So the maintenance of the active site for the enzyme requires a particular pH. And usually for most enzymes, that optimal pH is going to be anywhere from 6 to 8. But as you know, we have digestive enzymes that have to function in the stomach, and our stomach's pH is normally about pH 2. And so pepsin is an enzyme that you would find in the stomach, and you can tell that it would be the stomach enzyme by just looking at this graph here of pH scale. You can see the optimal pH of this particular enzyme is about 2.8 based on this graph. So you can tell that this enzyme is working in a very acidic environment, as opposed to trypsin, which is another digestive enzyme. Trypsin is found in the intestines, and the intestines are actually on the basic side. So the optimal pH for trypsin actually falls between 8 and 9. And then again, the average pH for, or optimal pH for enzymes in the human body is generally around 7. So the reason pH is a factor is because as you change pH, you're either adding or removing hydrogen protons. And this is going to disrupt the bonds, which will then disrupt the 3D shape of the enzyme. Disrupting the bonds, this is occurring through making changes or attractions between the charged amino acid R groups, which is going to affect both the secondary and tertiary structure. It's actually going to denature the protein because of those effects. And this again is what we're looking at in the graph, so I won't go over that again. The last condition is salinity or ionic conditions. And so as we change the ionic conditions, either too little or too much, we can have the same effect. This changes, it actually is adding or removing cations or anions, and which again is going to disrupt bonds. It's going to affect the secondary and tertiary structure. It can denature the protein, and it's going to have its disruption occurring between the R groups that have a charge to them, so the polar amino acids. An example, there are enzymes that can actually function at the edges of this, but there is a limit to the ionic conditions that any enzyme can function at. So the Dead Sea, which has a very high salinity, actually is called dead for a reason, because life can exist without the help of enzymes, and enzymes cannot exist in this extremely ionic solution. So the last part of this chapter is talking about little things that help the enzymes. They're little things, but they have a big effect. We either have inhibitors or activators that interact with enzymes. So the activators are things that are going to help the enzyme work. They can either be cofactors or coenzymes. So these are often non-protein helpers if they're cofactors. So for catalytic activity, these cofactors bind permanently or reversibly to an enzyme. And they're usually inorganic factors like zinc or iron or copper in an ionic form. If they happen to be organic cofactors, then we call them coenzymes. So things that are vitamins, like vitamin B3, niacin, riboflavin, coenzyme A, all of these are vitamins that act as coenzymes. Let's see, I'm trying to get on to our next. Oops, it's not letting me go to the right one. Okay. The other type of factor or compound that can affect enzyme activity is an inhibitor. And these are molecules that reduce the activity level of an enzyme. And they do it through one of these four ways: competitive inhibition, non-competitive inhibition, irreversible inhibition, and feedback inhibition. So competitive inhibitors are those that compete for the active site. So that's what defines that competitive part of inhibition. So for example, penicillin blocks enzymes that bacteria use to build cell walls. The penicillin actually binds to the active site, like in this picture here, so that the substrate cannot bind to the enzyme, and that kills the bacteria's functioning. So that's how penicillin works. And there are actually quite a few pharmaceuticals that work as competitive inhibitors. Disulfiram antabuse treats chronic alcoholism by blocking the enzyme that breaks down alcohol. So what that does is cause severe hangovers and vomiting, because if you have too much alcohol circulating in your bloodstream, it makes you really sick. It's a poison to our brains in particular. 5 to 10 minutes after drinking. So this particular competitive inhibitor acts to make alcohol a very negative experience. The only way you can overcome a competitive inhibitor is by increasing how much substrate is present in the system. So you can outcompete the competitive inhibitor by just adding more of the substrate. So if you saturate the solution, it will outcompete for the active site there. Okay, moving on. So non-competitive inhibitors. So these are not competing for the active site. And the reason is because they actually will bind to a totally different site on the enzyme. And when they do this, they cause a conformational change. That means they change the shape of the enzyme. So when they change the shape of the enzyme, they make the active site non-functional because they change its shape. So the substrate no longer will fit and bind to the active site. These are also called allosteric inhibitors because they bind to an allosteric site. So an allosteric site, think of allo as being other site. Allosteric sites are just not the active site of the enzyme. So it's called a non-competitive inhibitor can be an allosteric inhibitor. So again, we have a lot of pharmaceuticals using this type of inhibition. Anti-cancer drugs can inhibit enzymes involved in DNA synthesis so that you can stop the going crazy mitosis that's occurring in cells, the cancerous growth of a tumor. Another action of non-competitive inhibitors is with cyanide poisoning. And in this case, cyanide is fatal because it's irreversible as an inhibitor of cytochrome C. So cyanide will act as an allosteric inhibitor. And once it binds, it cannot be removed. So it totally inactivates these essential cytochrome C enzymes, which are involved in the production of ATP, which kills. It's a fatal poisoning. A lot of chemotherapy treatments use enzyme inhibition, and you can also control a lot of health disorders by selectively inhibiting different enzymes. So, like a common anti-cancer drug will stop the synthesis of a particular amino acid, which is involved in DNA synthesis. Okay, so I talked about cyanide as being irreversible. So both competitive and non-competitive inhibitors can permanently bind to an enzyme. If it's a competitive inhibitor that's irreversible, it permanently binds to the active site. If it's an allosteric inhibitor that's irreversible, it permanently binds to the allosteric site, which permanently will change the shape of the enzyme, making it inactive. Some other permanent allosteric inhibitors are nerve gas, sarin, which is another type of biologic warfare chemical, and most insecticides like malathion, parathion. These are all inhibitors that are involved in acetylcholine, which is a neurotransmitter's breakdown. So each enzyme has an active and inactive form. The binding of an activator stabilizes the active form of the enzyme, and the binding of an inhibitor stabilizes the inactive form of the enzyme. I'm going to pause for a second while my dog finishes barking. So in allosteric regulation, a protein's function at one site is affected by the binding of a regulatory molecule at a different site. This regulation can either be an inhibition or a stimulation of the enzyme's activity. So most allosterically regulated enzymes are constructed of two or more polypeptide chains. That means they have the quaternary structure. And so that complex that's formed, for example, here we have 4 different polypeptides joined together. That complex will oscillate between 2 shapes, one that's active and one that's inactive. And the one that's active is one that has an allosteric activator bonded to it, which stabilizes it. In a very simple case, an activating or inhibiting regulatory molecule will bind to a regulatory site. So it's not actually in the active site of the enzyme, it's actually a separate allosteric site. And the binding of that activator will stabilize the structure of the enzyme so that it stays with a functional active site. If it were the binding of an inhibitor at this spot, then we would have the opposite occurring. We would have it being stabilized into its non-functional form, where the active sites are not able to be accessed, or the substrate really can't bind to them. So pharmaceutical companies are actually spending a lot of time focusing on allosteric regulators that have a higher specificity for a specific enzyme than inhibitors that bind to the active site. Because an active site can be similar from one enzyme to another that's related, but allosteric regulatory sites evidently are very distinct between the different enzymes. So substrate, another form of regulation of enzymes, is when a substrate can act as an activator. So this is called cooperative or cooperativity, and it actually amplifies enzyme activity. So what happens with this is one substrate molecule, so remember that's the reactant in this enzyme reaction, will prime an enzyme to act on additional substrate molecules.